Hi everyone, my name is Daria and I'll be hosting the Computational Psychiatry and Development talks. So the first presentation is by Kailin Zhu. She's going to be talking about localizing excitation-inhibition imbalance in schizophrenia with virtual brains informed by white matter microstructure.
Thank you. So hello everyone, I'm an undergrad student from the University of Tübingen in Germany, and I'm very excited to talk about my work on the localization of excitation-inhibition imbalance in schizophrenia, especially using white matter microstructure. So 1 out of 3 people will have experienced some sort of hallucinations at some point in their life, which at least to me is very surprising. So these can be auditory verbal, or visual, or also other sensories. But yet only 1% of the population is diagnosed with chronic schizophrenia. So what is the difference here? So schizophrenia includes a broad range of symptoms. So these include delusions and also negative symptoms such as social withdrawal or impaired cognitive and emotional control. So now the challenge here is to provide early psychosis diagnosis and also hopefully to provide specialized treatment, so individually targeted at the root causes in every person, because in schizophrenia we have a high heterogeneity in the symptoms, so also really high into individual subject variability, as well as multi-domain nature of symptoms, since, which then require the localization of, for example, EI balance or any mechanistic disruptions that could be underlying these symptoms. So the hypothesis here is that in the brain we have an overall balance between excitation and inhibition, and that this could be disrupted and causing, for example, symptoms in schizophrenia. So we are looking at the cellular mechanisms. So this is EI balance and also the circuit mechanism. So we want to bridge it to the white matter microstructure. And so we want to explain the brain network differences in these symptoms. So how do we do that? How do we bridge the microscopic properties of EI balance to the whole brain and the brain activities? For that, we are using a virtual brain simulator. So we're using a whole brain simulation framework where we are setting up our model using a reduced Wong-Wong model. So every region in the brain is modeled by an excitation and an inhibition population. And so here we can see how these regions are modeled. So this is a stochastic differential equation about the firing rate and the synaptic gating variables that are informed by some structural connectivity weights. And the model itself is then divided into 2 different parts. So the first part is the feedback inhibition control. So here the goal is to settle it at a target firing rate. So we want to have that the average excitation and inhibition is on average at a target firing rate, that the brain is not exploding or the signals are not dying away. So the first part is just adjusting some regional parameters in every region that we call the JIs here, and they are adjusted according to a rule that is based on the learning rate and the difference between the synaptic gating variables and the target value. And the main part of the model is the EI tuning model. So here we are adjusting the parameters WFFI and WLRE, that is the feed-forward inhibition and the long-range excitation, respectively. And these are adjusted according to these 2 rules. So the model is informed by structural connectivity. So we have a matrix that shows the white matter connectome, so how every region of the brain is connected. And we then, the model then predicts functional connectivity. And in every step, the model is comparing the functional connectivity that is simulated and the empirical functional connectivity. And according to this difference and some loss function and a learning rate, the W parameters are adjusted in every step. So hopefully we can model the EI balance here. Now, does white matter matter? We want to know if the microstructure can actually have some influence for the EI balance. So right now in the literature, usually people would use the number and the density of fibers between 2 regions in the brain to inform the models. So we are using a data set here of 54 subjects, where 27 of them are schizophrenia, 27 are matched controls. And the table here shows the correlation value between the empirical functional connectivity per subject and the simulated functional connectivity. So ideally, we would want to have 1. Now, we can see that for number and density, it is around 0.2. But on the right side, we can see that for ADC and GFA, it is up to 0.7. So there's a drastic improvement there. Now, I hope you're wondering what ADC and GFA is. So ADC and GFA are ways to measure the fibers. So instead of taking the quantity, so the number and the density of fibers, we now want to see whether these fibers are actually aligned and directional. So ADC is the apparent diffusion coefficient that measures how freely water diffuses in tissue. And GFA is the generalized fractional anisotropy. So both of them measure whether the fibers in the brain are actually aligned. So we found that there is a drastic improvement in the model fit and the model performance by using ADC and GFA over number and density. So now we did some further analysis on that. We did some shuffling. So the first part, we did inter-individual shuffling. Surprisingly, here we found that apparently there is no significant difference in the model performance. So whether using my brain or any other brain here, the model is performing the same. So according to that, the white matter microstructure doesn't explain the dynamical differences in schizophrenia. However, we did more shuffling. So here we shuffled across the regions of the matrix, and that does seem to worsen the model performance significantly. So here we got a reduction of the R value from 0.7 to around 0.6. We did some further sparsity analysis where we used randomly generated graphs at different sparsity levels, and this also showed significantly worsened model performance as well as the best performance for a sparsity of around 0.7. That is the same as the empirical SC sparsity. So these are as expected. Now we did some visualization of the JI parameters. So here we can see the differences in control and schizophrenia and the top 10 regions where these differences are the largest for ADC and GFA. So the two regions that are in both of these lists is the posterior cingulate and the paracentral. So the paracentral is an associative sensory motor region, and the posterior cingulate is a region that is responsible for self-referential processes and social cognition. Now, are these EI maps useful for diagnostic classification? What can we do with these model parameters? So finally, we did some classification analysis where we're using a stochastic gradient descent and a random forest classifier, and we're informing these classifiers with the model parameters. So we're using the simulated JI and Ws to inform this into the classifier, and the task of the classifier is to hopefully predict whether this person is schizophrenia or control. Then we compared that to the same thing, but now informed with FC and SC, so only model-independent data. And we can see that both of them, especially the model parameters, they show that there's a huge improvement over chance. So with accuracy scores at around 0.7, it does seem to capture clinically relevant information. And also there's a slight improvement of the model parameters over the empirical data and the model-independent data. So now to conclude this, the main takeaways is that we're using a personalized whole brain model that is informed by white matter microstructure, and we showed that the performance for ADC and GFA outperforms the other metrics and the state-of-the-art number and density. As well as that, we found that the accuracy depends on the regional specificity, but not necessarily on the individual specificity of the white matter connectivity. And for diagnostics, we found that the classification indicates that the models can amplify clinically relevant dimensions and hopefully support the clinical diagnostics. And with that, I want to thank everyone in my lab for their great work, and especially to my supervisor, Chang-An, for the great support. And if you want more information, you can see our preprint with the QR code. And thank you all for your attention. Thanks to everyone for organizing CCN. Thank you.
We don't have time for questions, but you can speak to her after the session. So our next presenter is going to be Camilla van Geen. She is going to present distinct computational pathways to persistent fear across development.
Great. Hi, everyone. Thank you for still being here. I'm Camilla. I'm a postdoc in the NIV lab at Princeton. And yes, I'm going to be talking about distinct computational pathways to sustained fear across development. So we know that a core feature of anxiety disorders is unrelenting fear about an upcoming danger, even long after the actual threat has dissipated. And a successful approach to alleviating this fear is exposure therapy, which operates through a gradual process of extinction. And oftentimes, exposure therapy works well, but in about 50% of people, this extinction isn't lasting, and this fear is sustained or spontaneously returns just through the passage of time. And so our high-level goal here is to try to better understand why exactly this is happening so that we can then develop more targeted and hopefully more successful therapeutic interventions down the line. The first step towards this goal, we think, is to ask whether we can formalize a theory that explains this persistence of fear after threat has dissipated. And if so, can we then harness this theory to understand individual differences in threat response? And because anxiety is a disorder that's fundamentally developmental in nature, I'm going to be focusing here specifically on individual differences in development. So throughout the years, there have been several attempts at formalizing a cognitive theory of why fear may persist. But the framework that we believe explains the data best is latent cause inference. So the idea is that the world is governed by some latent organizing principles that give rise to specific and predictable outcomes. Importantly, though, these principles are not observable, and so they often need to be inferred. So you could imagine, for example, that every day I go to a local coffee shop and I get my coffee. Each day when I receive this coffee, it may look the same, but maybe behind the scenes, the actual quality of the coffee is changing according to the barista who was working that day. And so in order to correctly predict the outcome, which here is whether I will enjoy my coffee, I need to engage in a two-part process where not only do I have to infer which latent cause or which barista is active on any given day, but I also have to have learned to parse the environment into two relevant latent causes or baristas in the first place. And so the computational model here is essentially a parallel of this two-part process. Where on every trial, the agent first selects a prior, which corresponds to which cause it thinks is currently active. This is implemented through a Chinese restaurant process. The intuition here is that causes that have already had more trials assigned to them are more likely to be reinstated. And then with some probability that's set by the CRP prior, a new cause can also be created. Once the prior has been selected, the agent makes a prediction, and this corresponds to the expected value of the outcome given the cause that was selected. So in the model, this expectation corresponds to the mean of a beta distribution that's parameterized by that cause's observation counts. So to make that more concrete, let's say, for example, that I believe I'm in the cause where this yellow hoodie barista has made my coffee. And in the past, when that's been the case, the coffee has been good 3 times and bad 20 times. My prediction about the quality of the coffee would then be the mean of this beta distribution with 3 and 20 as its shape parameters. Then once the model has a prior and a prediction, it compares the prediction to the actual outcome and then uses Bayes' rule to compute a posterior, which here corresponds to the history of latent cause assignments that best predict the observed outcome. And so by the end of this process, you have information both about which cause explains which trial, but also about how many distinct causes best explain the full sequence of observations. So how can we actually apply this model to explain the persistence of fear? We do so using an online version of a fear conditioning and extinction task, where participants see 2 stimuli, a moon and a candle, And they learned that the moon is associated with a really aversive, high-pitched scream on 50% of trials. And the candle, on the other hand, is never paired with any outcome. Now, throughout the task, participants are periodically asked to make explicit ratings about how likely they think it is that each of the stimuli will be followed by a scream. So if they're learning correctly, their expectancy ratings during this phase of the task should look something like this. And then people enter an extinction phase in which the CS plus is never paired with the scream anymore. So if participants are learning, their expectancy ratings for the CS plus should drop. And then importantly, there's a 20-minute break with no new learning, no new exposure to the stimuli. And then we ask them again to predict whether each of the stimuli will be followed by a scream. So one way that we might see sustained fear looks something like this, where expectancy ratings for the CS minus stay low, but those for the CS plus spike again. Under the latent cause inference framework, this might happen if people are inferring 3 distinct latent causes, which I'm illustrating here. And then perhaps with time, memory of the neutral cause or association fades, while memory of the aversive cause, because it's more salient and valenced, is maintained in memory. And so the result here would be that when people are cued with the moon after the break, they'll report expecting a scream. All that being said, though, there's this idea of this neat partitioning into distinct latent causes that I think is a bit in tension with the fact that we also know that anxiety is linked to the overgeneralization of threat across cues. And so you could also imagine that threat would spread so that even latent causes that weren't originally predictive of an aversive outcome will start acquiring that link. And then you might expect that the expectancy ratings for the CS minus would follow those for the CS plus, and the behavioral profile would look something like this plot. And so we've identified 2 mechanisms with threat generalization and selective maintenance that both lead to a heightened threat response, but they have different behavioral signatures and different computational implications. And so now what I'm going to ask is whether thinking about these 2 distinct pathways can help us understand individual differences in fear learning across development. So there is some existing research that highlights both increased generalization and less lasting memory in children. And so with this in mind, we hypothesized that there might be an age-related shift that moves from generalization across latent causes in childhood towards the selective maintenance of aversive causes in young adulthood. And so with this trajectory in mind, let's see if this actually pans out in the behavior. So first, what I'm going to show is the proportion of people in each age group on the y-axis that belongs to each of these 2 behavioral subtypes, with people who show spontaneous recovery through the selective maintenance of the aversive latent cause, and then people who generalize threat across causes. And the assignments here were based on specific features of the dynamics of people's expectancy ratings. So first, in line with our predictions, we see that there are proportionally fewer children who show spontaneous recovery, so the red bar here is shorter than the yellow one. And then also we see that there are significantly more children who generalize. So now it's flipped and the red bar is higher than the yellow one. So finally, we can ask whether we can capture these differences as developmental shifts in parameters from our computational model of latent cause inference. So first, in line with the idea that latent causes may be less stable in children, we have a free parameter that controls what is essentially memory decay. And it's implemented by reducing the cause counts in the Chinese restaurant process, which makes old causes less likely to be reinstantiated. And the overgeneralization of threat, on the other hand, is implemented by modifying the observation counts themselves. So you could imagine that the prior has selected this latent cause, and then the outcome was a scream. There's a parameter in the model that basically determines the extent to which the scream observation also spreads to other latent causes that were not necessarily the ones that were selected on any given trial. And so we can look for age differences in the best fitting estimates of these parameters, which is what I'm plotting here. So age is now on the x-axis and the parameter values on the y-axis. And so higher values of gamma here actually indicate less decay, so more memory, just want to flag that. But broadly, what we see is in line with our predictions. So the younger children show more decay, reduced memory of existing latent causes, as indexed by lower gamma. And they also show, especially in this youngest age group, a tendency towards stronger generalization. And importantly, out of a total of 6 free parameters, these are the only 2 that show significant shifts with increasing age. So to conclude, we find evidence that children may form both less differentiated and less lasting latent causes. And in the context of fear learning, this means they show more diffuse, but also perhaps more temporary expectations of threat. And in the future, I'm interested in building up the idea that these biases could be normative, for instance, if children perceive their environment as more volatile. And of course, this could also vary across different people. So I'm also interested in relating the process to both early life adversity and mental health. And with that, I just want to say thank you to my collaborators, both at Princeton and NYU. And I'm out of time. Thank you for listening.
So the next talk is going to be by Chun-Hui Li. He's going to present the developmental trajectory of temporal dynamics in human visual representations.
Hi. Oh, okay. Hello, everyone. My name is Chun-Hui Li from Free University Berlin, and it's our honor to share our work on how human visual representations develop from infancy to adulthood. Previous EEG studies have shown that infant neuron responses already contain reliable information about the visual object. However, these studies have typically compared only a limited number of age cohorts, making it difficult to characterize the developmental trajectories across the lifespan. So, and more importantly, we still know relatively little about how the temporal dynamic and representational structure mature across the development. To address this gap, we collected a large cross-sectional developmental data set with 206 participants in 7 age cohorts, spanning from 6 months old to young adults. Participants viewed the 48 images from 4 categories: faces, objects, scenes, and body parts. During the experiment, participants will repeatedly present with these images. And infants and children viewed a subset of the stimulus images because of their shorter attention spans. Using this developmental data set, we ask how visual object representations develop across a broader developmental range. We approach this question from 3 complementary perspectives. First, their temporal dynamics, and second, the development of the category separation, and third, the latent representational patterns underlying these developmental changes. Let's begin with our first aspect, temporal dynamics. So we ask, how do the visual representations emerge, peak, and persistent? To investigate this question, we performed a pairwise image label decoding using a linear supported vector machine. And in the end, we averaged the decoding accuracy across all image pairs to obtain the decoding curve over time. Oh, sorry. The analysis revealed 3 clear developmental changes. First, image information becomes decodable at around 100 milliseconds and across all age cohorts. Moreover, the peak decoding accuracy increased with age, showing that image label representations become more discriminable across developments. The upper right panel shows how peak latency changed with age. Peak latency decreased most strongly early in life and became relatively stable, indicates that image-specific information becomes available sooner with increasing age. Finally, the temporal generalization matrices show how long the representations remain stable. Infants showed the relatively narrow and transient temporal generalization, while older children and adults show the broader off-diagonal generalization. Overall, visual representations become stronger, peak earlier, and persist longer with age. Next, we examined the category separation, in particular, how category structure develop across age. For each category, we first compared the within category decoding with the between category decoding. And we further defined the category separation as the difference between the two measures. Large value indicates the stronger category separation and the category refinement. We then examined how the category separation changed across the development. Here, the upper panels compare the within category and between category decoding across the 4 categories. In every category, the between category decoding were generally higher than within category decoding. This suggests that the broader category structure emerges early, but the exemplar representations continue to become more refined with age. Oh, sorry. The lower panels further show that this refinement was not uniform across the categories. Face information showed the strongest age-related change compared to the other categories. Therefore, category structure is present early, but both within category differentiation and the category separation continue to mature at different rates across the categories. Finally, we ask whether these developmental changes could be summarized by a small number of the latent patterns. Rather than analyzing each age cohort separately, we combine the time resolved RDNs from all participants into a tensor organized along the 3 dimensions: image pairs, time, and subject age. And we then applied the non-negative tensor decomposition to extract the latent pattern underlying the data. Each pattern can be characterized by the 3 loading profiles. First, the image pair loadings reveal which representational distinction define the pattern. And second, the temporal loadings show when the pattern is the most prominent. And the final one is the subject loadings, quantifying how strongly it is expressed in each subject. So in the result, we identified the 2 latent patterns with the distinct temporal and developmental profiles. The first pattern was characterized by the strong separation between scenes and the other categories, and it emerged relatively late, peaking at around 600 milliseconds, and its subject loading changed non-linearly with age. And the second pattern captured the broader separation among the visual categories. Its peak earlier at around 200 milliseconds, and its subject loadings increased strongly with age. Together, these results show that the visual development cannot be described as a uniform increase in representational strength. Instead, it involves the 2 distinct trajectories: a late scene-dominant pattern that changes non-linearly with age, and another early category separating pattern that becomes stronger across development. To conclude, our findings show the development of the visual object representations is multidimensional. First, visual representation becomes stronger and peak earlier and remain stable for longer with age. Second, different visual categories follow distinct developmental trajectories, and the category separation develop at the different rates across the visual categories. Finally, the tensor decomposition revealed the 2 latent representational patterns with different temporal and developmental profiles, and early category separating pattern and another late scene-dominant pattern. Together, these findings suggest that visual development is not simply a matter of the strongest representations. Rather, it involves a progressively reorganization of what information is represented and how it is structured across the development. So finally, I would like to thank you all of our team members, collaborators, and labor members, participants, and the funding agencies for their support throughout the work. Thank you very much.
Unfortunately, we again don't have time for questions. But our next speaker, Thibaut Chataing, is gonna present self-supervised modeling of social primitives in human dyads.
Good afternoon, everyone. I'm happy to be here and present you our research, human-Lisbeth self-supervised modeling of social primitive in human dyads, or more simply, feature engineering for social interaction from mice to human. So first, if we are talking about social interaction and we want to observe them, a model presented in 2023 proposed that the brain is first looking at three social primitives: interpersonal distance, so just the distance between the two people; facingness, the orientation between them; and contingent motion, which should be close to the synchronicity in their behaviors. So if we take that at the basics and then we want to observe social primitive and analyze them, we need some data for it. And if we want something more fine-grained than just some binary labelization, we can go to look at autism spectrum disorder. So in the definition of the World Health Organization, they say that one of the biggest symptoms of the autism is a degree of difficulty with social interaction. And that for us is interesting because autism is, for example, diagnostics with the ADOS, the Autism Diagnostic Observation Schedule, that give us, well, a binary label, so can be used as a proxy of social interaction or not, but that give us to see more fine-grained information that you will find in the diversity of the spectrum, for example. And we can also look at the Vinland, which is a complementary assessment that is more about everyday life skill. So we have those social primitives and the ADOS and Vinland that could be used as a proxy to measure social interaction, especially in young children where the communication is mostly nonverbal. So can we build an automatic model that will learn to see social interaction? So for that, we start on mice. So let me talk a little about Lisbeth, a transformer model that is currently in publication that take as inputs segments of preprocessing video of mice where we extract the pose estimation, so only the coordinate of the heads or tails, and will output an embedding that is optimized for social interaction, and later we can use clustering to detect, for example, attack, mounting, or investigation. So what we want to do is, well, take this model from mice and use it on humans. So for that, some change are needed. For example, the number of key points to describe the behavior. We need more key points for more degree of liberty of the human. But more importantly, well, we need to adapt to distinct behavioral configuration. So in more details, human Lisbeth, well, it's a transformers. It also takes as input the pose estimation, so coordinate of heads, hands of the two people in the scene. And then we'll project it to an embedding, and this embedding should have the information of the social interaction. But how that works? So just fastly, to do that, we worked on data that are recording of ADOS sessions, so dyadic of clinician and children's for the ADOS assessment, so the autistic diagnosis. So the training of the model works with self-supervised tasks. So the idea is we project the records in the new embedding, and this embedding, we will challenge it to see if it presents the information that we are interested in. For the core task, we have 4 different challenges. The group consistency, it's a binary challenge, so we take either an original sample or we modify it. The modification is, if we take a scene that is shaking hands, for example, of 2 people, well, we will replace one of the 2 individuals with an individual from another scene, which is, for example, 2 people jumping. And you can imagine this new scene of one people is checking hand in the void and one in, jumping next to it. It completely destroys the meaning, and we want the models to be able to see that, to be able to predict that this scene has been altered. So the order, temporal order, we take the segment, we cut it, and switch, like breaking the causality. For the temporal shift, what we do is we take one of the two individuals and we shift it in time. So if we take the example of the shaking hands, well, when one of the individual is still shaking the hand, the other is already walking away. And for the last, the temporal warp is just spinning up or slowing down the segment. So just to summarize on that, what we are doing here is we challenge the embedding to see if it is able to answer any of those 4 questions. And if it's a success or a failure, that will look back on the training. That's how the model will optimize itself. Then we have the last task, the spatial invariance, that is more to decode human social scenes. So this one has a little different in the paradigm. It's a contrastive learning process. So we have the original sample. Let's take the shaking hands again, and then we create positive sample from it by using translation, mirror in the x-axis, or a zooming process. So it gave us multiple positive samples, and then we sample negative samples. It's other behaviors like the jumping or whatever you can imagine. And we put all of that in the embedding, and we tell him, okay, the original and positive samples should be close together in the embedding. The rest, you can put it far away. What does that mean? It means that in the embedding, if you are shaking hand in front of the camera or in the back of the room, well, the model will create the same representation of it. So it means it will learn relational geometry, not absolute position. So with this learning process, we want to dig deeper and to see how, what information human Lisbeth is learning. And for that, we compare it to 2 baselines, one which is a kinematic null model. We just compute speed, acceleration on the pose estimation, some kinematics, and another one where we add on those kinematics some social metrics, so a proxy for interpersonal distance, facingness, and congruent motion. And the target here are, well, the ADOS information that we talked about earlier. So the diagnosis to see if we have autistic information, but also the more fine-grained details that will give us more insight about what kind of information are found in the embedding. So to do that, we need to aggregate the features because we can't do the prediction by each frames. We need to do them over some time scales so they can match the diagnosis. So what do we get? First, all of the 3 models managed to predict the diagnosis. Not amazingly, but good enough so that we can say, okay, there is information in it, so we can dig deeper. And then what we are interested in is the differentiation. So are a model significantly better than the other? And that's what we found is, well, 14 types. 14 where we can't say it's significantly better or worse, but we also find that for Vinland personal and Vinland community, human Lisbeth is better. So it means a win on the relational and real-world information. That is more the type of Vinland. But on the other hand, there is a lose on the temporal contingency and the non-dyadic. And that is about the ADOS social affect or severity score, but both are highly correlated together, and the Vinland domestic, which is non-dyadic. But concretely, what does it tell us? Well, it means the human Lisbeths tend to capture the relational geometry, and that tends to go with the spatial invariance task on the learning process that I showed earlier. But it meets in temporal contingency, and that is more difficult to conclude on it because we have a strong aggregation in the task, so that can be an explanation of why we lose that much temporal information in it. So to go deeper on it, what we want to do to understand more of what this model is learning is first an evaluation study where we can just try the mice model on human and see what can happen. Then we want to take out the segment level pooling to analyze better what the temporal information are staying in it. Trying a decoding probe where we want to try to predict from the embedding directly the 3 social primitives to see if they survive in the information or if the model is looking at something else. And finally, try it on 3D relational geometry because, well, 2D has some bias that may limit the information found into the model. And thank you for all the lab and for your attention for the talk.
Thank you. Does anyone have any questions? Okay. Well, I have a quick question. Do you think that you'll actually be able to decode phasiness and from the latent embeddings of the model? You haven't tried to do any preliminary?
No, we have not tried yet for the decoding, but from what we do in analyzing the behavior that are found in the autistic children, because we use it over all the records, is yes, we tend to get the relational geometry where, for example, the two people are really close together or far apart at the distinction of the behavior. So at least interpersonal distance, phasiness is more difficult to do because more difficult to define in 2D. So that's, we need to dig deeper to see.
Okay, thank you so much. Let's thank him again. So our next presenter is Domenic Bersch. He's going to present vision models that capture complementary representations in children that converge in adults.
Thank you so much. Hello, everyone. My name is Domenic Bersch, and I am a third-year PhD student at the Goethe University of Frankfurt. And the paper that we have submitted claims that vision models capture complementary representations in children that converge in adults. And once I received the message that I have the honor to present here today, I panicked and set a goal for myself to find out what these representations are. So in the name of development, this talk will be about the development of the visual system, development of my method, and the development of me aging 10 years in the course of the last 2 weeks. Well, let's start from the beginning. So high-level visual representations change well into adulthood. And for example, an 8-year-old doesn't represent this room as you do, and over the course of a decade, this will change as well. And while model brain alignment has already been heavily studied in adults, the developmental aspect is still underrepresented. And this part isn't only relevant from a neuroscientific perspective, but also from the development of AI models. And this is what our lab at CVAI is interested in. That's our logo, by the way. First off, what is the data we are working with? So we are working with the same data as my colleague Chun-Hui, who presented 2 slides before me. We have a total of 48 images from 4 categories, being faces, objects, scenes, and body parts. These have been viewed by multiple subjects of ages 8-year-old, 12-year-olds, and adults with EEG recorded. For the analysis in the session, we built a 48 by 48 RDM for each subject for every pair of images to show how differently the brain responds to them. Okay, so now what is the perspective of our lab and why do we want to bring AI models in? It's no news that AI and cognitive neuroscience have a symbiosis, like advancing one might help advancing the other. And we want to harness the symbiosis from a developmental perspective. How do we learn? How do we change? How could the model learn, or how could the model change? In other words, can we use the mechanisms that we find in developing brain to advance the learning paradigm in AI models? And this all would happen under the term of continual learning. And this is basically the motivation our lab has. Okay, going back to our abstract submission, what was the finding? So in this plot, you can see the average alignment of different ANNs across different age groups, across time. First panel shows 8-year-olds, second shows 12-year-olds, and then the adult age group. Then the reddish models are ANNs that have been trained to classify images with visual input only, including different architectures like CNNs, vision transformer, different training paradigms like self-supervised or supervised. And the blue model is CLIP, a transformer architecture that has been trained on images but with language supervision. The pattern I want you to focus on is that CLIP seems to be able to explain brain alignment in children, while the other models only seem to converge in adults. And this raises the question, what does CLIP have, what the other models do not? And this is also the part where I aged 10 years, because I was trying to find the exact mechanism for this talk, but let's see how far I got. For now, let's focus on CLIP and vision transformer, because they're both transformers and better comparable. And since they are both transformers, they overlap a lot. And to understand where they differ, I need to isolate the part that's unique to CLIP. So to achieve that, I take all layer RDMs of a vision transformer and use them to predict one layer of CLIP by linear fit. That predicted part is then what both models share, because if vision transformer can predict it, it's because they share it. Then to get CLIP's unique part, I just subtract the shared part from CLIP RDM and continue with my EEG correlations with that. Okay, so this plot is the exact same as we saw earlier. We're looking at CLIP in blue and vision transformer in red. For both models, we're looking at layer 11. The reason for that is that as vision transformers are already very similar to CLIP, we need to look at the layer where they diverge the most to really ask what is unique to CLIP, and that's at layer 11. As a note, 2 of those 12 vision transformer layers do have slight significance in 8-year-olds, but CLIP is significant in 10 out of 12 layers. We want to find the mechanism that really drives this pattern, so we're taking this layer. So now what happens if we remove all of vision transformer from CLIP and vice versa? Okay, what do we see here? The thick lines are the unique parts of the model. The thin lines are what the full model used to be. First off, removing all of vision transformer from CLIP still keeps CLIP significant across all age groups. So whatever drives the correlation here is unique to CLIP. Second, removing all of CLIP from vision transformer leads to it not being significant in adults anymore, which is fair since CLIP seems to add more to the brain model alignment compared to vision transformer alone. One thing that I found interesting is that the absolute alignment of CLIP unique doesn't grow across age group. It actually drops a little bit in adults. So that means whatever makes CLIP unique seems to be a stable scaffolding, which is necessary to explain representations in children. So yeah, we're looking at a constant, something that's already at adult strength in 8-year-olds, but still something that's only captured by CLIP. Okay, so if this part doesn't develop, then what does? So to answer this, what develops across age, we can drop the models for a second and only look at the EEG alone. We created 2 binary RDMs, same versus different category and living versus non-living. And what we did is we correlated each hypothesis RDM with every subject average across the defined time window below. And here we can see that the alignment roughly doubles from 8-year-olds to adults. So these semantic distinctions seem to sharpen with age, which is not surprising. Representation sharpens well into adulthood. But bringing back CLIP into the picture, gray is the normal alignment, remove these 2 RDMs and the correlation drops, which is again also expected, but the alignment also stops growing. So CLIP's developmental part is the semantic part, and that is also the part that it shares with the vision transformer. But this still leaves the other half open. What exactly is the constant scaffolding that only CLIP has that's able to explain children EEG? So first, an obvious question would be if we can really associate language supervision to be the driving factor. And for that, I was looking at 2 models from the SLIP paper, CLIP and SimClear, this time both trained on the same data set, same parameters, same EPOS, and so on. Here looking at layer 7 for the same reasons as before. SimClear trains by contrasting images against other images. We can see that in red, and CLIP is the same as before, contrasting images against text, only dataset match this time. All right, what can we see here? Similar pattern as before. Also here, CLIP is significant across all age groups. This time, only SimClear seems to be also significant across all age groups. Okay, so now if the alignment is really driven by language supervision, removing all of SimClear should keep CLIP positive in children, right? And it seems like a yes. CLIP is significant in children, and SimClear goes flat and only remains significant in adults. Is that our story? It's tempting to say yes, since many points point towards this direction, but SimClear also has augmentation as part of its objective, so we cannot really isolate the paradigm here as well. So I was hoping to have that unique mechanism prepared for today, but sadly I didn't manage yet. However, what we do know is that CLIP survives the removal of all of vision transformer and all of SimClear and remains significant across all age groups, specifically in children. That unique part is not the part that increases into adulthood. It seems to be like a stable scaffolding. And the part that is growing and changing into adulthood is the sharpening of the semantics. And this is where the other models seem to align with. Overall, my or our lab's main goal is not to examine only CLIP, obviously, but rather to make sense of all of the mechanisms in AI that drive alignment across different age groups. And we are already working with mechanistic applications, feature steering, and more importantly, also an increased data pool with ages ranging from 12 months to adults. And yeah, this will all happen in constant communication with other labs, especially labs with more knowledge on the neuroscientific end, to make sure that we make sense of our findings and yeah, explore models that learn like the developing brain rather than just ending up like the adult one. That being said, hopefully cool things to come. Let's see how things develop, pun intended. Until then, thank you for listening, and see you around.
Thank you. Does anyone have any questions?
Hi.
Hello.
I have a question for you. Given that you have EEG data, which part of the brain are you recording to? And when you're aligning the visual representation, what part of the brain are you measuring?
So for these results, it was the entire brain region, so all of the channels available. I already tried some experiments with a reduced fraction of the channels, but I ran into noise issues, so my results ran really significant when I tried this. I think there are methods to overcome this, but I haven't figured it out yet, sadly. So that's why I've been using whole brain EEG for now.
Okay, thank you.
Thank you for your question. Okay, bye.
Thank you again. Our next and final presenter is Elizabeth Im, and she is going to present on the topic, Do You See What I See? Mobile eye tracking in natural environments reveals selective social visual engagement and developing visual field biases in children and teens.
Sorry, I'm having a hard time making this full screen. That's not... Thank you. Thank you for being here for the last talk of the last session. So as we develop between childhood and teenage years, we learn many new things and do different things. For example, here is 4-year-old Leah. Her room is full of toys, she's playing with Legos, her parents are also reading her picture books, and she's learning how to grip a pencil. Here's teenage Leah. She now spends time sitting at her desk, doing homework, and she's so good at typing that she doesn't even have to look at the keyboard. She FaceTimes her friends, and she's learning how to drive. Between these periods of time, both what Leah does and her environment changes. How do these developmental changes impact her visual experience? Our goal is to understand children's visual learning curriculum. Researchers have long hypothesized that our visual diet, which is the information they're exposed to and attend to throughout everyday life, shapes how the brain develops and how we learn about the visual world. Characterizing this helps us understand the inputs that drive visual learning, provides insight into the brain development, and can inform the development of more biologically plausible models of visual development. To do this, we focused on 3 aspects of children's visual diet. First, the prevalence of different visual categories, which is how often they appear in the visual input. Frequent exposure to a category may influence how well it is learned and represented. Second, where we fixate, because where we fixate better reflects what we attend to in our messy real-world visual environments. Comparing prevalence and fixation helps us understand not only what information is available, but also what children fixate and prioritize in their daily life. Finally, we're interested in the visual field biases, where different categories tend to appear. Consistent spatial regularities, such as faces appearing near the center of gaze, may contribute to the organization of the developing visual cortex. However, what is children and teens' visual diet in natural environments and how it changes is largely unknown, because up to recently, there was no technology that enabled measuring fixation patterns in natural environments. Today's talk aims to fill this gap by asking what is children and teens' visual diet in natural environments and how does it change during development? While there's many things in our visual diet, for the purpose of today's talk, I will focus on 2 categories, faces and hands, for 2 reasons. First, in the ventral temporal cortex, which is involved in processing and categorizing higher-level visual information, we find that from age 5 to 17, face selectivity increases and limb selectivity decreases. This finding raises the possibility that increased looking at faces and decreased looking at limbs over development may drive this neural change. Second, in VTC, face-selective regions have a consistent foveal bias, and limb-selective regions have a consistent lower visual field bias. It has been hypothesized that because face recognition requires high visual acuity afforded by foveal vision, fixation on faces generates the foveal bias on faces. As faces and limbs are attached to our body, this fixation pattern puts limbs consistently in the lower visual field. These fixation patterns have only been measured in lab settings and viewings of photos and mostly been characterized in older teens and adults. Is this fixation pattern how young children also see other people in their daily life? While these studies allow us to posit possible hypotheses about how children's visual diet of faces and hands may change or not change, This has largely been speculative, and we don't know which parts of our visual diet are linked to brain development. To answer this question, we use Pupil Lab mobile eye tracking glasses to measure scene and fixation patterns in children ages 4 to 7 and teens 14 to 17 at home, and children ages 4 to 5 in school while they do their everyday activities. But in the interest of time, I'll be focusing on home data today. We first tested the accuracy of the device during free viewing. Children and adults were asked to fixate at 12 locations spanning a room, and we calculated the error between target and the eye tracker fixation point. This shows the error for 10 children and 10 adults for each of the fixation target. The error was about 1 degree, and there was no difference between groups, suggesting that we can resolve fixation on a face up to about 11 meters away. First, to measure prevalence, we use AI-based methods by combining 2 vision models, YOLO Everything and MMPose, to automatically detect all faces and hands in the videos. Our performance is high, both detections reaching over the strong performance F1 threshold of 0.7. To give you a gist of the data, here are randomly sampled frames in which faces were detected, and you can see the model is doing pretty well. And here's example frames from each age group for hand detection. And before I show you, I want to note that for the rest of the talk, I will use orange for children and blue for teens. We first quantify how prevalent the faces and hands in their environment. We find that in home, children, 40% of the frames contain faces, but in teens, only 20% of the frames contain faces. In contrast, in both children and teens, 60% of the frames contain hands. Then we asked, do they spend time looking at faces and hands differently? Interestingly, when we quantified the percentage of frames on which face is fixated upon, we find that teens fixate on faces 20% of the time, but children only 12% of the time. So even while faces are less prevalent in teens' environments, they're more likely to fixate on them than children. We find no differences on fixation on hands. We also measured the eccentricity, which is the distance from fixation of faces and hands. On the x-axis, we have eccentricity in degrees, where 0 is closest to fovea. On the y-axis, we have proportion of frames with faces or hands of that eccentricity. For faces, we find that for both children and teens, some faces are fixated on, and many faces are in the periphery. And in general, children's visual diet consists of significantly more faces in the periphery than teens. And eccentricity of hands are similar. Lastly, we compared the distribution of upper-lower visual field bias relative to fixation and asked, do children and teens have different visual field biases? On the y-axis, we have vertical bias, where 0 is center of gaze, and above 0 is above gaze center, and below 0 is below gaze center. On the x-axis, we have percentage of frames without vertical bias for faces and hands. We find that for children, faces more often appear in the upper visual field. However, for teens, faces are centered along the line of gaze, not showing visual field bias. We also find visual field differences for hands. Hands are more often in the lower visual field for teens, but hands are at the center of gaze in children. At home environments, visual field biases for hands and faces differ between age groups. Today, we started by identifying a gap. To fill this gap, we collected visual diet data and tested standing hypotheses in the field about the developing visual diet. One hypothesis was that between childhood and teenage years, we will have more faces and less hands in our visual diet. We found that face prevalence actually goes down and hand prevalence remains similar. However, what did increase was fixation behaviors. Teens had higher fixation percentage on faces that were present in their visual diet. We also examined visual field biases in children's natural settings. We find that visual field biases are developing in young children. There are more peripheral faces in children's visual diet, and hands appear more below center of gaze in teens, but not children. Existing theories about why category selective regions emerge where they do in VTC posits that is driven by fixation behaviors. However, these theories are mainly built on adult data or in lab settings, and assumes we look at natural environments the same across development. We find that children and teens have differing social visual engagement strategies, and visual field biases in the natural world are changing from childhood to teenage years, suggesting the possibility that visual field biases in the visual cortex will also develop during childhood. In our ongoing work, we continue to characterize this rich data set to better understand the developing visual diet. Thank you for listening.
Thank you, Elizabeth. Does anyone have any quick questions? Yeah, I guess, why do you think that teenagers fixate on faces more, even though they appear less in their environments?
I think there are a couple reasons why. I think one reason could be that as you grow older, you're better at understanding what face information conveys, like emotional cues, gaze information. And so that means for teenagers, faces contain more information that they can extract. And so, but to extract those information, you probably have to look a little bit more. Also for children, in our visual diet, there's many, like millions of things. Like there's chair, there's ground, there's screens, there's so many things. But maybe for teens at home environment, they're already used to, this pen, is the same as this pen. So maybe they don't spend as much time exploring different categories of information. And so for teens, the thing that is moving and is conveying most interesting information, maybe the faces. But in our ongoing analysis, we're trying to better understand that.
Thank you so much. Let's thank all the speakers. Thank you also for being here. And this concludes the last contributed talk session for CCN. Thank you.